I have always had an interest in the natural world, and it was sometime during my early university studies that I became curious of how nature functions and the molecular mechanisms of life's inner workings and chemistry. I got the opportunity to volunteer at a research station in the rainforest of northeastern Australia. There was bioluminescent fungi, huge blue iridescent butterflies, and incredible plant biodiversity. And I think this trip was one of the formative experiences that then led to the research path that I have followed. My name is Totte Niittylä, and I'm a professor in plant physiology and department head for forest genetics and plant physiology at SLU. My research group investigates plant growth, and we focus on trees and the formation of wood. We're trying to understand what goes on inside the trees when they take up carbon dioxide from the atmosphere and turn it into wood. And in our work, we identify and study the genes and enzymes required in this process. We're especially interested in wood because it's one of the largest renewable resources that we have on the planet, and it's also a major carbon dioxide sink, which is important for mitigation and adaptation to climate change. And wood also it provides the largest renewable resource for raw materials and reduces our dependency on petrochemicals. In our work, we use aspen as a model system for trees, and we also work on the little model plant Arabidopsis, which we use to study more specific fundamental questions related to cell biology. Trees are slow. They are a bit like ants in The Lord of the Rings, slow to respond to change, but once they respond, it can go fast. So we're studying trees to understand how they distribute and prioritize their carbon reserves between growth and storage, and how climate change affects these processes. I believe a better understanding of tree biology can contribute to healthier people and planet. In the big picture, I would like to contribute to knowledge required to build more sustainable societies.
Again, a warm welcome.
Thank you.
In the video, you mentioned that a better understanding of tree biology could contribute to a healthier planet and healthier people. Could you elaborate a little on how you see that connection?
Yeah, I think so. Trees they're incredibly important in many ways for us and to our societies, and they contribute to cleaner air and cool down our cities, and they are stopping erosion, and they provide, as I mentioned in the video, the largest resource of renewable materials. So the list is very long, and as tree biologists, we're trying to understand how trees function and grow. So by understanding that you can sort of assist the trees, and which will then also, you can maybe say that if trees are doing well, then we are doing well also. There's this sort of interplay.
Yeah, thank you. And in your research, you follow carbon from the atmosphere to the moment it becomes wood. Do you have a sense of how long that journey takes from the moment a tree absorbs a carbon dioxide molecule through its stomata in a leaf or needle, and to when the carbon is finally incorporated into the wood?
Yeah, we do have quite a good understanding of the timings of that. So it depends a little bit of the size and the age of the tree. So in the young saplings that we work a lot in the lab and in the greenhouses, it's a matter of hours. While in the bigger trees and in the forest, it can take days or even weeks, and in some extreme cases even years. When depending a little bit what path the carbon takes, and that's some of the questions that we're studying in the laboratory is the different pathways that the carbon can choose from.
I think you will come back to that a bit. And before you begin your talk, a more personal question. Was there a moment or discovery during your early career that was particularly influential in leading you to pursue a career in science?
Yeah, that's actually something I've been thinking about now in coming up to this day and this event, in that you sort of reflect, okay, how what's the path that I have taken, and you can sort of define some moments in the research career that have been important. And if I would, from the early days, single out one, it was during my PhD work. We were trying to understand how plants survive through the night, and what kind of carbon reserves they use and how they use it. And we'd identified this interesting mutant in this model plant Arabidopsis that seemed to provide some of the answers, but we didn't know where the mutation was. And at that time, mapping the location of the mutation in the genome would take, if everything went well, it would take about 1 year. Now you can do it in weeks or a month or so. But the DNA sequencing technologies at that time meant that it would take longer, and I remember this moment in the lab where I'd been sort of trying to map this mutation, and maybe 8 months, 9 months down the line, and I got some sequencing results in the lab, and there was only me and one other colleague left in the evening, and then I finding this mutation and seeing there on the screen, and you couldn't sort of believe it, because you until then you always had this like, okay, am I on the right path, or am I slightly off, or you have to go back and start again. And then it was also fun because it was the mutation turned out to be in a gene which was a little bit unexpected, and the discovery then led to some changes in the textbooks in plant biology that then was sort of a nice jumping stone to jumping step to the career, and in a way sort of you realize that I started to realize that maybe you could make a living out of the working in this field actually, and here we are.
Yeah, and how did you celebrate that moment?
I think we went for I did my PhD in the UK, so we went for a pint.
Okay.
I think
of course. Oh, exciting. Well, it's time for you to get ready for your lecture, and I would like to so please take the stage. I would like to introduce Totte Niittylä, Professor of Plant Physiology, and he is going to talk about following the carbon from the atmosphere to the wood.
Yeah, thank you for the introduction. So yes, my title of my talk, Following the Carbon from the Atmosphere to Wood, summarizes the kind of work that we focus on in my laboratory. And what we're doing is to identifying genes and enzymes that are important in this process. But to make sure that everybody is on the sort of right page, I start with this introduction to photosynthesis. So, as you probably all are aware, plants have this ability to utilize the energy from the sun to capture energy from the sun and take up carbon dioxide and water and turn it into sugars and oxygen in this process called photosynthesis. What is perhaps less appreciated is illustrated on the picture to the right, and that is that about 1/7 of the atmospheric carbon dioxide passes through this process annually on a global scale, which means that in 7 years, all of the carbon dioxide in the atmosphere has been through photosynthesis. So this gives you some idea of the scale of this carbon flux on the sort of on the planetary context. And so I think against that background, it's interesting to look at this graph that is also familiar to most of us, the increase of atmospheric CO2 since the start of the industrialization. So starting here from the mid-17th century, pre-industrial times, and we have data from ice cores showing the steady but slow increase of carbon dioxide in the atmosphere, and then things start picking up after the Second World War, and since from 1950s, 1958 to be exact, we have more high-resolution temporal data of carbon dioxide increase in the atmosphere. And if we focus on that latter part of the graph, we can see this that there is this sort of saw-line pattern in the carbon dioxide changes that seems to follow an annual pattern. And the peak is always in May, so it's right now, and then the atmospheric CO2 drops during the summer, and the explanation is like in front of our eyes. If you go outside now, is what is happening in the northern hemisphere is that the leaves are flushing, and the photosynthesis is really kicking off, and the planet is kind of breathing in now a lot of carbon dioxide. And this map sort of illustrates where it is happening. So the green areas are forests, and the darker the green, the stronger carbon sink the forested area is. And the northern hemisphere simply contains more of the green landmass that is responsible for this phenomena of carbon uptake. And getting to the topic in my laboratory is the fact that most of the carbon in the forests is found in wood. So really there is this close interplay between trees and how our world is behaving, and understanding the process of carbon uptake and then how the carbon is after photosynthesis turned to wood is important also at the planetary scale. So how does it work then? So one of the key molecules in this process after photosynthesis is sucrose, which is the common white sugar that we're all familiar with and use in our kitchen. Because this is the molecule that plants redistribute the carbon resources. And sucrose is transported up and down the tree stem. And if we focus in using a light microscope and look at the tissue where this transport happens, we're here on the right-hand side seeing this tissue called phloem, which is the tissue where the sucrose is transported long distances along the stem. And then from the phloem, in order to get to the wood and wood-forming zone, it crosses radially into the wood across this tissue called cambium, which is where the sort of stem cells are that are dividing and producing phloem to the outside and wood to the inside. And my lab has been over the past years working a lot on this process of how the sucrose is transported from the phloem across cambium into the wood, and then how it is metabolized. So how the sucrose is broken down initially before the carbon from the sucrose is turned into wood, and the process of wood formation. The other important question that we have been investigating is like how do trees then prioritize their carbon use? So if one simplifies, the tree has two possibilities after photosynthesis. It can either store the carbon for future use, and most plants store carbon as starch, illustrated here in this picture as a chloroplast which has starch granules inside it. And starch is it's an important energy reserve for plants, but also, of course, the main source of calories for us humans. Alternatively to the storage, the tree can invest into growth, and so it's a balance between storage and growth that is the sort of question for the system, and how this regulation happens it's open for. It's not fully understood, so we've been investigating this process. And we use these plants as our model systems. So our main model system for tree biology is aspen, shown here. The reasons being that it has a relatively small genome. It was the first tree genome to be sequenced. There are lots of sort of molecular biology tools that we can use to address sort of questions in aspen as a model system. And then if we want to move faster, we choose Arabidopsis, and especially for questions that are well conserved between plants. So we can learn many things in Arabidopsis that are transferable to trees, but not always. So there are also many questions that you only can study in trees. So there's often good arguments that we cannot use Arabidopsis, but we have to do our experiments in aspen. And the third model system that we use is spruce, which is arguably the most important plant in Sweden, both economically and ecologically, or in the Nordic countries. But of course, spruce grows slower. It has a giant genome, and we don't have the same sort of molecular tools to ask the kind of questions that we can do in Arabidopsis and aspen. But for some specific questions, the spruce is also a very good model system. In our work, an important tool are these gene editing tools, which allow us to modify specific pathways or specific processes in plants. So here one example is where we recently asked the question of the importance of the storage processes in trees, and we basically stop the trees from storing carbon, stop them from making starch. And we created these starchless aspen trees, shown here on the right. And the assay that you can do for starch is using iodine to stain leaves, and iodine stains starch black. So you can see here in the wild type, the leaf disc taken from these plants is stains completely black, while the starchless aspens, they remain brown. And to our surprise, these trees grew very well. And this was a bit unexpected because the same kind of mutant in Arabidopsis is very small and sick. So this was a good example again that of kind of work that models that we learn in Arabidopsis cannot be just directly always transferred to trees. So we need to study trees in order to really understand some basic fundamental questions. We often use our automated tree phenotyping facility in Umeå, which you also saw in the video. So this is a sort of great system to get very high resolution growth data, how the trees are growing. And basically the trees are moving on these conveyor belts in the system, and they go through this white box here at the back where we have different cameras measuring different parameters. So we get leaf area, we get height and stem thickness, we also get leaf temperature, and many different things. So it allows to get very sort of nice data on how the trees are growing. And from this facility, we take the most interesting findings to the field to then test that how do the trees cope with real life outside. And this is our field experimental site located in the next to the SLU campus in Alnarp. And this is a good example of why SLU is an excellent place to do this kind of work. So we have our greenhouses and laboratories in Umeå, but we can run experiments in the south where our trees are doing much better. They grow faster, and they even have a nice sea view where they sort of are so kind of opportunity to do work in the field is important for testing the models that sometimes in sort of the lab-based models and how they transfer to a real life. So I mentioned that we're following the carbon in my title, and this slide tries to illustrate what I mean by it. So you can make this analogy between traffic flows and metabolic flux, or the flow of carbon. So often metabolic pathways are depicted as sort of roadmap-like systems. So we have different metabolites that flow through different biochemical enzymatic reactions catalyzed by enzymes, and similarly you have sort of traffic maps, and the traffic is flowing through the system, and then there are places where the traffic gets jammed, and similarly there are places in metabolism where the flow of carbon gets jammed or slows down. And with our experiments we can manipulate these pathways. So what we're often doing is that we maybe deleting a pathway like we did with the starchless mutants, or we can add a new sort of bypass and then study like how does the system adapt to it and what happens to the flow of carbon when we do those kind of changes. And that can tell us something about the fundamental mechanisms of how trees grow and how they respond to different changes or stresses, for example. The way to follow the carbon, though, is we cannot set cameras in the pathway, but we're using an isotope of carbon called carbon-13. Carbon-13, we feed it into the system as 13CO2, and this labels the metabolites with this carbon-13 isotope, which we can then track in the system. So we can sort of run an experiment where we put the trees in an atmosphere containing 13 CO2, and then we run a time course where we sort of follow how long does it take for the carbon to get from A to B, for example, and which pathway does it take. And sometimes we're learning surprising things when we do this. So we can, for example, what we have sort of found, for example, is that we find things that there is a pathway that is not on our map. And that illustrates sort of one of the important questions in this field is that there are a lot of unmapped areas in sort of plant metabolism, and some of them unmapped areas are clearly very important. So we are trying to understand which enzymes and genes are responsible for those unmapped areas of the metabolic pathways. This point also leads to a sort of second project, which is maybe not one. It's not the main focus in our lab, but something that sometimes I have a postdoc or a student coming to the group that is sort of who wants to do something novel and new and has this sort of lust for discovery and new areas, and this is where a project that we designed some years ago to take on some of the challenge of the unknown gene space, as we call it. So approximately still 1/3 of the about 28,000 genes in the best studied model plant Arabidopsis are still of unknown function, even though we've studied this plant for decades now. Tens of thousands of scientists have spent time on identifying genes and their functions. So we're still 1/3 in the dark. And to make inroads into this it's quite tough work. That's why it's taking such a long time. And we started with a project where we're sort of breaking new ground by identifying previously uncharacterized essential plant cell processes. And that's where this is an experiment or work where we work with the sort of very early stages of Arabidopsis development at seedling or even embryo stage. So we're looking at mutations which kill the plants before they even really get started. So that's like a sort of hallmark of, okay, the process is essential. And from this work, we've identified a number of interesting leads. One of the main leads that we ended up following, I had first a picture on this, but then the picture became very complicated. So against the usual presentation rules, I try to sort of summarize this now in this sentence, which summarizes our findings of one of the projects coming out of this work, is that we basically identified the new protein complex that we call opener. And this is a complex that works on both the cell's control center, the nucleus, and its energy center, which is mitochondria, to build ribosomes. And these ribosomes, they're sort of molecular machines that make proteins. And proteins are they're the enzymes that make, for example, carbon move and things happen in biology. So this is also something that we're not following further in my group, I don't think, but the person who was sort of pioneering this work has now moved on, and they will continue this work on their own. So it's given a jumping stone for somebody else to get into a research career. And you may wonder, how does this link to wood formation? Then sounds very interesting. But there is ribosomes in wood as well, and actually they play a very important role in wood formation. In that we see this sort of wave of ribosomes, which I've tried to illustrate, ribosome biosynthesis, which I've tried to illustrate here with this white line in the cambium zone, and then stretching out to the wood forming zone. Basically the ribosome, there's a sort of massive investment into making ribosomes before you can make wood, simply because ribosomes make the enzymes and the proteins that are required for wood formation. So our opener complex is involved, is an important critical component of this ribosome biosynthesis process. Okay, so with that, I will wrap up and just like to emphasize how I see research as very much as a team effort. And if you prompt generative AI to draw a picture of research as a team effort, you get this kind of a cute heart-shaped image with many cells, which I think illustrate all the different collaborators that I have had the pleasure of working with from different cultures, different countries, lots of many bright minds. And my role maybe has been just to be the sort of the epidermis to kind of keep things together and make sure that people stay focused and enthusiastic about their work. So thank you for your attention, and happy to take some questions.
So welcome back to your seat here. Yeah, thank you very much, Totte, for an interesting lecture. We have time for one or two questions. Are there any questions from the audience? Yes, Pernilla, please.
Thank you, Totte, for a great lecture. When the amount of carbon bound in wood increases, what trade-off do trees make to be able to absorb and bind more carbon dioxide, and how does it affect their life cycle span? And do they become more sensitive to insect attacks, need more water? What kind of trade-off?
Yeah. So the usually the reserve carbon, which is the other option, in a way that if the tree doesn't invest into growth and it puts into reserves, that's kind of the tree's insurance policy, in that they can use those reserves to defend itself against different kinds of stresses. So there is for a tree breeder, for example, if you want to make a tree that grows faster at the expense of reducing the storage capacity, then you run a risk of making a more vulnerable tree, for sure. So it depends a little bit. It may be a reasonable approach for a short rotation forestry, with the time scales of where you can sort of have a more riskier growth approach, and in return you have a faster growth.
Okay. Well, if we manage to understand how wood is formed, how might that knowledge be applied in the future, you say?
I think we have some interesting collaborations with people working on global vegetation models and climate modeling. They're telling us that there is this black box on how, for example, the storage to growth division happens. So understanding that process might give us better understanding of how trees and tree growth affect climate. And then, of course, tree breeders, which often identify variants of different genes that contribute to better growth. What we can provide is understanding of what those genes are actually doing. And so that can give added confidence to a tree breeder that, okay, we know the function of these trees and understand how they increase the growth. So let's make this cross or introduce this particular gene variant to this tree.
Yeah. I'm afraid we don't have more time for more questions, but we will give you a warm applause. Thank you very much, Totte.
Yeah.